Hi, my name is Arya Shah, and I'm going to be talking about the Parkinson's disease and how the Parkin protein is activated by PINK1 gene. So, the Parkinson's disease can be caused by mutations in the Parkin, also called systolic ubiquitin E3 ligase and the PINK1 genes, which disrupt mitophagy and the destruction of the damaged mitochondria. Parkin is an E3 ubiquitin ligase that normally tags mitochondria for degradation. So the degradation is a process by which proteins are naturally destroyed in the cell in order to maintain protein homeostasis, or an equilibrium of proteins in the human body. However, Parkin is autoinhibited by its ubiquitin domain, blocking the enzyme active site. PINK1 activates Parkin through a two-step mechanism, first phosphorylating the ubiquitin, then phosphorylating Parkin ubiquitin domain. So phosphorylation is the addition of a phosphate group to a protein or other organic molecule. It is an important mechanism for regulating protein function and activity. Then, also, while ubiquitin phosphorylation is critical for Parkin activation, the precise structural rearrangements involved are unknown. Elucidating the conformational changes underlying Parkin activation by PINK1 is key to understanding the Parkinson's pathogenesis and designing new therapies. In Figure 1, you can see the experiments were done on a garter snake Parkin, which is similar to human Parkin. Parkin is a protein involved in the Parkinson's disease. The experiment shown that when the garter snake Parkin is activated, it undergoes similar changes to the human Parkin. One, the ubiquitin domain, which is part of Parkin, becomes phosphorylated, where the phosphate groups attach to it. Then the linker region between the ubiquitin domain and the UPD domain becomes more structural and ordered. The ring 2 domain, another part of Parkin, is exposed and released from the UPD domain. The goal of this project was to determine the mechanism of Parkin activation by PINK1 phosphorylation, and that we hypothesized that the phosphorylation of Parkin ubiquitin domain by PINK1 includes the conformational changes that releases the ring 2 domain from its uninhibited position obstructed by the UPD domain, thereby activating Parkin and enabling its ubiquitin ligase activity. Specifically, the Parkin is a protein involved in the Parkinson's disease, as I've mentioned above. It has several domains, including the UBL domain, Ring 2 domain, and UPD domain. Usually, the Ring 2 and UPD domains are bound together, which keeps the Parkin in an inactive site. The UBL domain activates as the activation switch. When the UBL domain gets phosphorylated, it causes a change in the shape and structure of the Parkin. Specifically, the phosphorylation of UBL leads to ring 2 and UPD domains separating from each other. This releases the ring 2 domain and activates Parkin. In the Parkinson's disease, there are mutations in the Parkin that prevent this activation process from working properly. The mutations block UBL phosphorylation so that the ring 2 and UPD interactions is not released. Understanding the molecular details of how UBL phosphorylation activates Parkin gives scientists targets to design activators. These activators can mimic UBL phosphorylation to force the ring 2 and UPD domains apart, contradicting the effects of Parkinson's mutations. This could restore the normal Parkin activation in patients. The methods: hydrogen deuterium exchange mass spectrometry, which basically tracks down changes in the Parkin when it's activated by phosphorylation. It also looks at the amide hydrogen and the protein backbone. These hydrogens can be exchanged with deuterium, which is a heavy hydrogen from water. Areas that become more exposed when Parkin is activated will exchange hydrogen faster. Areas that become more protected will exchange slower by monitoring the exchange over time with MS, which is a mix mass spectrometry. It maps out regions of Parkin that changes exposure upon activation. It also determines the 3D structure of activated Parkin found to phosphorylate ubiquitin. Also, there's another method which was used in this research, was Parkin activity assays. These measure the Parkin is active by looking at its ubiquitin conjunction ability. A ubiquitin probe that labels Parkin's cysteine is used to see if Parkin is is an open and active state. Ubiquitination assays mix parkin with E1 and E2 and ubiquitin enzymes. Formation of the ubiquitin chains and parkin shows it as active. Activity is measured by dic- dictating ubiquitin chains on parkins using the Western blotting. As you can see in figure 2a, this shows a crystal figure of the human parkin. The core part of this parkin was crystallized. This includes the UPD, ring 1, IBR, and UBL domains. The ring 2 domain is also included, which is bound to the core. The phosphorubiquitin domain is not present in the structure, but is located as shown in the sign dash shape. The ubiquitin domain and ubiquitin has phosphates attached to serine 65 residues highlighted in the structure. This cartoon shows the different domains and is crystallized together by UPD, ring 1, IBR, and phosphorubiquitin and the bound ring of R2 domain. In figure 2, you can see that it shows an open book view of the interaction between the Parkin UPD domain and the ring 2 domain. The UPD domain and the R2 domain normally bind together, which keeps Parkin in an autoinhibited or a closed state. This interaction surface is hydrophobic, meaning it involves nonpolar contracts with the other two domains. When parking gets activated, its ubiquitin domain to phosphorylate ubiquitin, this releases the ubiquitin domain from the core of the parking. However, this figure illustrates that even when ubiquitin binds to phosphoubiquitin, the ring 2 domain remains bound to the UPD throughout the same hydrophobic interface. So the autoinhibitory ring 2 UPD interaction is still intact. This implies that ubiquitin phosphorylation alone is not enough to fully activate the parkin. The ring 2 domain needs to be released from the UPD to open up parkin's catalytic site. Additional steps are likely needed to break down ring 2 UPD interface. This may involve further phosphorylation of the structural rearrangements that disrupt the hydrophilic contacts. Next, the figures and the results show that when phosphorylation ubiquitin binds to the parkin, it releases the UBL domain, but the key inhibitory parkin regions stay inbound. In contrast, when UBL is phosphorylated by the pink one, the REP and the ring 2 domains get exposed over time. This suggests that the ring 2 domains start detached from the parking core. The phosphorylation UBL that rebinds to the UPD domain, this fully releases the ring 2 domain, making the parking active. The crystal structure shows in phosphorubiquitin binds to the UPD using hydrophobic patch, putting a PSIR65 into pocket lined by key residues mutated in the Parkinson's. This is also conserved as an ACT linker between UBL and UPD. It shields UPD when ring 2 is released. ACT residues, leucine 102, valine 105, and leucine 107 mimic the hydrophobic ring 2 residues as MET 458, TRIP 462, and PHEN 463. Mutating the ACT sequence prevents the activation shown in the which is crucial for the release in the ring 2 phosphorubiquitin. In figure 3, this shows that the linker region between Parkin ubiquitin domain and UPD domain. This within the linker is the ACT element, which consists of two serine phosphorylated sites, serine 101 and serine 108. The structure shows the ACT element with SIR 101 and SIR 108 resolved, meaning the positions have been seen. Phosphorylation of the SIR 101 was found to decrease parkin activity. This is likely because the adding of the phosphate to the SIR 101 prevents the phosphorubiquitin domain or the linker from binding to UPD. So SIR 101 phosphorylation provides a regulatory switch to inhibit parkin activity when needed. So in conclusion, the parkin is an E3 ubiquitin ligase involved in mitophagy and it's linked to the Parkinson's disease. Let's walk through how phosphorylation activates parkin using the recent structural studies. Parkin is kept in an inactive state by binding to its catalytic ring 2 domain to a regulatory PUPD domain. Phosphorylation induces major shape changes that reveal a hydrogen deuterium exchange mass spec. The critical change in this ring 2 domain gets a release from the UPD. This frees a Parkin catalytic site so it interacts with the E3 enzymes and ubiquitin substrates. Highly resolution crystal structures allows us to visualize exactly how phosphorylation leads to the ring 2 release at a molecular level. Phosphorylation occurs on a Parkin's ubiquitin domain at serine 65 by this upstream kinase PINK1. This phosphorubiquitin then binds to the pocket of the UPD domain. In this pocket, phosphorserine 65 makes key contact with the residues lysine 161, arginine 163, and lysine 211. Disease mutations at these sites prevent activation. When phosphorubiquitin binds to UPD, it mimics the normal interaction of the ring 2 domain. This causes ring 2 to detach from the binding site of UPD, and an ACT linker between UBL and UPD domains also contributes important hydrophobic contracts with the stabilized the active state. With ring 2 releases, it can now freely move and interact with E3 enzymes. It swings over a 550A from its inhibited position. And I want to acknowledge the diamond light source with scientists and collections. And lastly, I want to thank Dr. Russell for helping me through this process. Thank you so much.